Staphylococcus aureus. Staphylo means grape shape. Coccus means round. Aureus means golden color. Staphylococcus aureus is a gram-positive, round-shaped bacterium, and it is a member of the common microbiota of the human body. Frequently found in the upper respiratory tract and on the skin. It is a facultative anaerobe that can grow without the need for oxygen. Although Staphylococcus aureus usually acts as a commensal of the human microbiota, it can also become an opportunistic pathogen, being a common cause of skin infections, including abscesses, respiratory infections such as sinusitis, and food poisoning. Pathogenic strains often promote infections by producing virulence factors, such as potent protein toxins, and the expression of a cell surface protein that binds and inactivates antibodies. The emergence of antibiotic-resistant Staphylococcus aureus, MRSA, is a worldwide clinical problem. Despite much research and development, no vaccine for Staphylococcus aureus has been approved. An estimated 20% to 30% of the human population are long-term carriers Staphylococcus aureus, which can be found as part of the normal skin flora, in the nostrils, and as a normal inhabitant of the lower reproductive tract of women. Staphylococcus aureus can cause a range of illnesses, from minor skin infections, such as pimples, impetigo, cellulitis, folliculitis, carbuncles, scalded skin syndrome, and abscesses, to life-threatening diseases, such as pneumonia, meningitis, osteomyelitis, endocarditis, toxic shock syndrome, bacteremia, and sepsis. It is still one of the 5 most common causes of hospital-acquired infections, and is often the cause of wound infections following surgery. Each year, around 500,000 patients in hospitals of the United States contract a staphylococcal infection, chiefly by Staphylococcus aureus. Up to 50,000 deaths each year in the USA are linked with Staphylococcus aureus infections. 1881, Sir Alexander Ogston, a Scottish surgeon, discovered that Staphylococcus can cause wound infections, after noticing groups of bacteria in pus from a surgical abscess during a procedure he was performing. He named it Staphylococcus, after its clustered appearance evident under a microscope. Then, in 1884, German scientist Friedrich Julius Rosenbach identified Staphylococcus aureus, discriminating and separating it from Staphylococcus albus, a related bacterium. In the early 1930s, doctors began to use a more streamlined test to detect the presence of a Staphylococcus aureus infection by the means of coagulase testing, which enables detection of an enzyme produced by the bacterium. Prior to the 1940s, Staphylococcus aureus infections were fatal in the majority of patients. However, doctors discovered that the use of penicillin could cure Staphylococcus aureus infections. Unfortunately, by the end of the 1940s, penicillin resistance became widespread amongst this bacterium population, and outbreaks of the resistant strain began to occur. Staphylococcus aureus can be sorted into 10 dominant human lineages. There are numerous minor lineages as well, but these are not seen in the population as often. Only a few strains of Staphylococcus aureus are associated with infections in humans. Another notable evolutionary process within the Staphylococcus aureus species is its coevolution with its human hosts. Over time, this parasitic relationship has led to the bacterium's ability to be carried in the nasopharynx of humans without causing symptoms or infection. This allows it to be passed throughout the human population, increasing its fitness as a species. However, only approximately 50% of the human population are carriers of Staphylococcus aureus, with 20% as continuous carriers and 30% as intermittent. This leads scientists to believe that there are many factors that determine whether Staphylococcus aureus is carried asymptomatically in humans, including factors that are specific to an individual person. According to a 1995 study by Hoffman et al., these factors may include age, sex, diabetes, and smoking. They also determined some genetic variations in humans that lead to an increased ability for Staphylococcus aureus to colonize. In conclusion, there is evidence that any strain of this bacterium can become invasive, as this is highly dependent upon human factors. Microbiology. Key characteristics of Staphylococcus aureus. Staphylococcus aureus. Grape cluster berry. Aureus, golden. It is a facultative anaerobic gram-positive cockle, round bacterium also known as golden staph. Staphylococcus aureus is non-motile and does not form spores. In medical literature, the bacterium is often referred to as Staph aureus. Staphylococcus aureus appears as staphylococci, grape-like clusters, when viewed through a microscope, and has large, round, golden yellow colonies, often with hemolysis, when grown on blood agar plates. Staphylococcus aureus reproduces asexually by binary fission. Complete separation of the daughter cells is mediated by Staphylococcus aureus autolysin, and in its absence or targeted inhibition, the daughter cells remain attached to one another and appear as clusters. Staphylococcus aureus is catalase positive, meaning it can produce the enzyme catalase. Catalase converts hydrogen peroxide to water and oxygen. Catalase activity tests are sometimes used to distinguish staphylococci from enterococci and streptococci. This is the end about Staphylococcus aureus. Thanks for watching. If you like, please thumb up and leave a message. You must be my biggest motivation to make the video. Please subscribe to the channel, where you'll find better medical videos later.